All right, so chapter 6 is going to be all about the organelles that make up a cell. But we're going to start by just discussing what cells are and what their importance is in organisms. Cells are the base unit that makes up all of our organisms. Some organisms are unicellular, many organisms are multicellular, but it is the simplest collection of matter that we can have and still have a living thing. Cell structure is going to be related to their function, and cells are going to come from previous cells. So there's some examples of some more specialized cells that we have in our bodies. We just did our lab with microscopes, and microscopes, this chapter section of the book is going to focus on that. There isn't so much of this to cover on the reading guide, but I did want to talk about it just briefly in case you wanted some more information about it. So we can't see cells typically just by our eyes, but the microscopes will allow us to visualize them by magnifying them and allow us to, with resolution, to be able to see the components that make up our cells. So light microscopes allow visible light to move through your specimen and then into the glass lens. The lenses are able to bend the light, which is able to enhance the image in terms of its magnification. So magnification is comparing the object's actual size to that we see through as opposed to its real size. Resolution is dealing with the clarity. So the image on the left in the middle is going to be have a greater resolution than the one on the right. On the one on the left, you can see the individual indentations on this dog's nose. And then contrast is going to be able to show you specific differences in parts of the sample, the difference between the darker areas and your lighter areas. So just to kind of give you some context as opposed to size, we can see that most plant and animal cells are going to fall between 10 and 100 micrometers, and those can be visualized using a light microscope. When we start to get into organelles, they're anywhere from 1 to 10 micrometers. As we continue to get even smaller, we tend to have to use either super resolution microscopy or electron microscopy to be able to see individual ribosomes, viruses, proteins, things like that. If you want to be able to see something that does not require a microscope, you can use, you basically need it to have a size approximately, I would say 50 micrometers or greater. Human eggs fall just kind of outside of unaided eye, but frog eggs, nerve cell lengths, muscle cell lengths, obviously our height, anything like that we can see without using a microscope. And if you want to see individual molecules or atoms, you're going to have to go even further than electron microscopy. The light microscopes typically can magnify up to about 1,000 times the size of the actual specimen. We can stain our slides with the cells that are present to make it easier to visualize them. And most of your subcellular structures, your organelles, aren't going to be as easy to identify using a light microscope. You might be able to see the nucleus, but that might be about it. So there are advances that have occurred in microscopy that have helped to be able to see things in greater dimensions, 3D viewings, and you'll see some pictures as we go throughout the text where they show you different views of components that make up organisms that have been enhanced by more advanced microscopy techniques. And then obviously labeling will also help to make it easier to visualize, and that's what you're seeing down there in that bottom picture. It's the same image, it's just that the one has been labeled as opposed to the other. So electron microscopes are able to look at subcellular components. Specifically, scanning electron microscopes will take a beam of electrons and push them onto a specimen surface, and by doing that, it's able to provide what appears to be 3D images. Transmission electron microscopes take a beam of electrons and put it through a specimen so that you can examine its internal structure. If we look at cell fractionation, that can actually break cells apart and then separate organelles from one another so that we can identify what their functions are. So it's able to, again, by depending on the mass, be able to spread things out even further and further. And so between this type of analysis, cytology, as well as biochemistry, we can determine how the structure of the different organelles correlates with their function. Section 2, eukaryotic cells are going to have internal membranes that are able to compartmentalize their functions. Our cells in every organism are either going to be prokaryotic or eukaryotic. If you are a part of the bacteria or archaea domain, then you are going to consist of prokaryotic cells. If you are part of the eukaryota domain, your fungi, animals, and plant kingdoms, as well as your protists, you're going to consist of eukaryotic cells. So what do these cell types have in common? They both have plasma membrane. They have the cytoplasm, known as a cytosol. They have chromosomes that are able to carry genes that will code for proteins, and they have ribosomes, which are what are actually able to make the proteins. But prokaryotic cells do not have a specific nucleus. Their DNA is located in a generalized region called the nucleoid. They have no membrane-bound organelles, and the cytoplasm is kept intact by that plasma membrane. And what you see there is an example of a rod-shaped bacterium. While eukaryotic cells have more structure to them, their DNA is housed in a membrane-bound envelope, the nuclear membrane. They have membrane-bound organelles. Their cytoplasm basically separates the plasma membrane from the nucleus, and they tend to be a lot larger. So the plasma membrane is basically acting as the gatekeeper for what enters and exits the cell, depending on what the composition is of what wants to enter and leave. Some things are able to move through it passively, other things require assistance, and some require more than just assistance, they require energy. And we'll spend more time on that in our next chapter. But the general structure that we see with our biological membranes, our plasma membranes, is a double layer of phospholipids, also known as a phospholipid bilayer. And we spent some time recently talking about hydrophilic and hydrophobic regions. Again, there's going to be a hydrophilic region outside of the membrane as well as inside in your cells. And so the phospho part of the phospholipid bilayer is facing those more hydrophilic regions, the ones that are facing the aqueous environment, while the lipid components that make up the tails of the phospholipid bilayer face one another so that they are facing away from the aqueous environments. So cells can only really get to a certain size because as they get to a certain size, that's going to make it harder for them to meet their requirements metabolically to do what they need to do in order to function. So as their membrane space decreases per volume, as they get bigger, it's harder for them to get everything they need. So the smaller a cell is, the more surface area that membrane has, which allows it to both take in nutrients that it needs as well as remove waste. So you're not likely to see cells in that middle column. You're more likely to see lots of those smaller cells that you see in the first column making up roughly the same space as the middle column. So I know that you have studied cells before. There is a eukaryotic cell breakdown, some things that are unique to your eukaryotic cells, and we're going to talk about each of these, so I'm not going to spend as much time on this right now. We have flagellum on some of our cells. We have a pretty enhanced cytoskeleton. And we do not have large vacuoles. Although I'm looking and I'm not sure if I see a vacuole per se on here. I'm not sure why they don't have a vacuole on here. That's probably not kosher. But this is actually from your text, so that's something for them to add on. But I do want you to notice that the nuclear membrane is basically surrounded by your endoplasmic reticulum. And then your endoplasmic reticulum basically falls right next to your Golgi apparatus. And so the RNA that moves out of your nucleus is going to go straight to either a free ribosome or the ribosomes that are found on your ER. And as the proteins are made from the amino acids joining together, they can then be processed and packaged and sent off on their merry way using the Golgi apparatus. Well, with a plant cell, we see that there are two layers to making up the exterior of your cell. You have not just a plasma membrane, but you also have a cell wall. You see much larger vacuoles, or you should see more. There we go. There's your central vacuole right there. Sorry, I'm being silly. And you still have your nucleus very close to the ER, and you have the Golgi apparatus pretty close by as well. The other organelle that we do not see in our animal cells that is present is your chloroplast. So that's going to be a special feature just to your plant eukaryotic cells. So we're going to focus first on the nucleus and how we use the information that we take out of the nucleus and get it to the ribosomes to make our proteins. And that is our genetic material, which is going to be able to determine what the cell needs to do. So the majority of a cell's DNA is found in its nucleus, and the ribosomes take the information that the mRNA brings it from the DNA to make the proteins that the cell needs. So your nucleus is basically your control center of your cell. It's got the majority of your cell's genes. Remember that there is some DNA found in your mitochondria as well. The nuclear envelope basically separates the nucleus from the cytoplasm that surrounds it. It is a double membrane and it is a lipid bilayer. There is a nucleolus found inside of that nuclear membrane, inside of the nucleus, and that is where ribosomal RNA is able to be synthesized. So you can see your nucleolus kind of there in the center. You see the pores, which are going to be one way materials can enter and exit the nucleus. It's also how you can get your RNA out. And then you can see the chromatin that has been bound together when that cell is not going through the process of replication. So as I said, pores are going to regulate what enters and exits. How the nucleus is able to maintain its shape is what's called the nuclear lamina, and it's basically just a framework. We'll spend a little bit more time talking about that when we get into the different filaments that help to provide structure and support for the cell. But the nuclear lamina is primarily made of protein. So within the nucleus, you're going to have your chromosomes. The chromosomes are DNA that is associated with proteins. They get wound together and make chromatin. And when it is ready to be duplicated, when the DNA is ready to be duplicated, the chromatin will condense and form those specific chromosomes. But as you can see, you've got your double helix there, the nucleosomes that have the protein on them. The DNA gets wound onto them, and it gets wound as well onto histones, and those together make up your chromatin fibers, and then all the chromatin fibers help to make up your chromosomes. So ribosomes are where the proteins are going to get made. They are made of both ribosomal RNA as well as protein. So you're going to need protein synthesis to take place in one of two places, either in the cytoplasm on ribosomes that are just free out in that aqueous environment, or you're going to have it take place along the endoplasmic reticulum or the nuclear envelope if there are bound ribosomes present. And we will spend a lot more time talking about replication and transcription and translation later on this year. So the endomembrane system is what is able to help to get the pieces that make up your protein and then the proteins themselves from one point to another. So it's basically your factory. So you can have components be transferred between these either continuously, or they can be moved between different components using vesicles. So you're going to go from your nuclear envelope to your ER to your Golgi apparatus, and then depending on what the function is, it could go to a lysosome, it could go to a vacuole, or it could be headed towards a plasma membrane. So the ER is going to take up or make up more than half of the total membrane that's present in most eukaryotic cells. As I said, it is continuous with your nuclear envelope. There are two components to it. You can have a component that is called smooth ER that does not have ribosomes present, so that would be where there is not protein synthesis occurring. And then you have the rough ER, where you do have protein synthesis taking place. So why would you need to have smooth ER versus rough ER? Smooth ER is not responsible for protein synthesis, but it is responsible for lipid synthesis. And keep in mind that your membranes are made of lots and lots of lipids, so you definitely need a place to make lipids in your cells. It also will help to break down carbohydrates. It helps to detoxify any drugs or poisons that might enter, and it's also a source of calcium ions. Your rough ER, again, is going to be your primary source of protein synthesis. It's able to take the message from the mRNA and translate it into amino acids and then join them together to make your peptides. It is a source of transport vesicles that is able to carry proteins, among other things. And it's a membrane factory and helps to provide compartmentalization throughout the cell. Your Golgi apparatus is able to take those proteins that have been transported either directly or through vesicles and is able to help to move them through that to get them sent to where they need to go. So the Golgi apparatus is a series of flattened membranous sacs. They're called cisternae, and they have a cis phase and they have a trans phase. They are able to fold and modify proteins that are made through the ER. They can make some macromolecules, and they're able to act as a postal office. They're able to sort and package materials so they can go where they need to be headed. There are tags on them to help to identify them. Lysosomes are a means to break things down. They contain, it's a sac of hydrolytic enzymes that can break down your 4 macromolecules. It tends to work better in an acidic environment, and so that's what you see present in your lysosome. So they help to degrade molecules that are either worn out or damaged or just no longer needed. So one way they can do this is by engulfing other cells, also known as phagocytosis. And when they go through this process, they basically are producing a food vacuole. And when the lysosome fuses with it, it's able to break down the different molecules inside of it. As I said, they can also break down worn out macromolecules. This includes your organelles as well. And that particular process is called autophagy. And then they can also help to destroy a cell if it's no longer working properly. Programmed cell death, also called apoptosis. So there you can see a lysosome that has acquired a food vacuole and is bringing that in. And you can also see a lysosome helping to break down organelles that are no longer functioning properly. Again, it could be macromolecules, anything along those lines. Vacuoles are going to be a source of lots of different materials. Your plant cells tend to have much larger vacuoles than other eukaryotic cells. The vacuoles are derived in part from your ER and in part from your Golgi. They tend to, when you have a food vacuole, that tends to form as a result of phagocytosis. Your contractile vacuoles, which we see often in freshwater protists, are a way to remove water. And then central vacuoles, which we see in plant cells, they don't just hold on to water, they can also hold on to organic compounds. So there's an example of your plant vacuole. So again, just as a quick refresher, the nucleus has your DNA, which is transcribed into your mRNA, which then leaves through the nuclear pores and goes to the ER to produce proteins, at least the rough side of the ER, or to free ribosomes. And then those proteins are either transferred directly or they're vesicles to the Golgi, and they move from the cis side of the Golgi to the trans side, where they are processed and then shipped off to their final destination, or at least the destination that they next need to head off to. So mitochondria and chloroplasts, we're going to spend a couple of chapters talking about their functions with aerobic respiration and anaerobic respiration, as well as photosynthesis. But these are ways that the cells are able to get energy. Mitochondria are present in both plant and animal eukaryotic cells. They are where cellular respiration occurs, and when it is done aerobically, that is when you're going to get the most bang for your buck. That's when you're going to get the best use of the materials that you are consuming to make ATP. Chloroplasts will only be found in your plant and algae cells, and that is where your cells are able to take carbon dioxide and water and use them to make sugar. And they also produce a lot of oxygen. We also have another type of organelle, a perioxisome, which is just an oxidative organelle. So how did mitochondria and chloroplasts come to be? These actually have double membranes as well, and there are some similarities to them with bacteria because they do have their own DNA. I said earlier our mitochondria have DNA in animal cells, but chloroplasts also have DNA molecules. So plant cells have two sources of DNA besides the nucleus. They also have free ribosomes, so they can do protein synthesis all on their own. They tend to be able to grow and reproduce a little bit more independently within the cells. So there's definitely been some studies as to how these came to be, why these organelles have these different properties compared to other organelles. So the theory that is thought to have led to the development of mitochondria and chloroplasts is called the endosymbiont theory. And it's thought that early ancestors of eukaryotic cells to produce a mitochondrion accepted or engulfed a non-photosynthetic prokaryotic cell, and rather than breaking it down, it formed a relationship with it, and it allowed that particular cell to to reside within it. And as that cell duplicated itself, it became a eukaryotic cell with that organelle, the mitochondrion. It's also thought for the chloroplast to have developed that one of these early ancestors engulfed a photosynthetic prokaryote, and that was basically thought to be the start of plant cells. Okay, so a prokaryote was engulfed, one that was non-photosynthetic, the other one that was photosynthetic, and as they replicated, we now had eukaryotic cells that either contained chloroplast chloroplast and mitochondrion or just mitochondrion. So again, mitochondria are what give you your energy. They have, again, that double membrane. They have an outer membrane and then an inner folded membrane, which maximizes their surface area. That inner membrane can results in having some space between the two membranes, the intermembrane space, and it also creates a matrix, which is what's inside the folded area. And there are some components of cellular respiration that can only take place within that matrix. Again, because you have those folds, the cristae, there are lots of places for enzymes that are going to be able to make ATP to be present. And so that's part of the reason that the mitochondria is such an efficient converter of ADP into ATP, your energy currency. Chloroplasts contain chlorophyll and other enzymes that help with photosynthesis. These tend to be found in your leaves and green organs of your plants as well as algae. And if you look at the structure of your chloroplasts, you see that they have these stacks of thylakoids. They're called thylakoid membranes. And then the thylakoid membranes are surrounded by stroma in the inner membrane, and then you also have the outer membrane on the outside. If you have a set of thylakoids together, you have a granum, and those are what are going to allow the light and dark reactions to occur in photosynthesis. And so this just happens to be one of your plant organelles that's known as a plastid. So I said we were going to talk a little bit more about peroxisomes. These, again, are specialized metabolic compartments, so that's why we're talking about them and result with energy, because if we metabolize, we're going to generate some energy. These are actually only bound by a single membrane. They are able to take hydrogen peroxide and turn it into water, and we still have a lot more to look at within the peroxisomes, but because they are able to do those oxidative processes, They are able to conduct a lot of different reactions that have different functions. When you can oxidize, you can do all sorts of stuff. So cytoskeleton. These are fibers that help to organize the cell structure as well as activities that take place within the cells. They're basically the roadmap for your cell. They extend throughout the cytoplasm, and there are three main types that are going to play a role: your microtubules, your microfilaments, and your intermediate filaments. So what does the cytoskeleton help with? It helps to support the cell, it helps to keep its shape, it helps it be able to move with the assistance of motor proteins, and it's able to, again, act as your roadmap. It's able to create monorails that the vesicles can travel along. It's also thought that it may play a role with regulation of biochemical activities that take place within your cell. So the fibers that make up your cytoskeleton include your microtubules, your microfilaments, and your intermediate filaments. Your microtubules are the thickest, your microfilaments are the thinnest, also known as actin, and the intermediates fall between the two. So that's just going over the different sizes that you see with those three. I know we're going to spend a little bit more time on them. So with the microtubules, they, again, help more with motion. They are able to help chromosomes move during cell division. They're able to help your organelles move, and they help to maintain cell shape. They're the compression resistors. The microfilaments are able to bear things, their tension. They're able to help the cell change shape a little bit, and that's something that you might need with cell division. They're able to assist with muscle contraction, with the process of cytoplasm streaming, and we're going to see this when we do our lab with photosynthesis. They're able to help a little bit with movement, like with pseudopods, and they can help with forming that cleavage furrow that needs to take place in cell division. Your intermediate filaments also help with the cell shape, with the tension bearing. They are part of that nuclear lamina, and they help to anchor the nucleus as well as other organelles so that everything that needs to work together is going to be close enough to be able to do that. So again, we talked a little bit about each of those on that previous slide. The microtubules are able to move organelles as they need to be moved and help to maintain the cell shape and help to separate chromosomes from one another. How they're able to do that, they reach out from a centrosome that we only find in our animal eukaryotic cells. That is also known as an MTOC, a microtubule organizing center. These centrioles the centrosome consists of a pair of centrioles that have 9 triplets of microtubules that are basically arranged in a ring, and so they can reach out and grab onto all the different chromosomes that are present. They also help cilia and flagella beat. Cilia and flagella do not beat the same way. The microtubules are basically helping to make up that cilia and flagella. A basal body is what anchors it in place, and this motor protein dynein is what determines whether they bend, going back and forth, or whether they're able to physically move in one direction versus another. So with the cilia, again, we can see them kind of moving back and forth. With the flagella, we see more direct motion take place. So what is able to make this occur? The dynein protein basically grabs on and then moves the microtubules and then releases them. But because it's grabbing onto them basically one at a time, and that they are linked by multiple proteins, it prevents them from sliding off and not being controlled by the dynein protein. And it can cause the cilium or the flagellum to bend as a result. Your microfilaments are again going to be involved with generating the cortex, which exists just inside of your plasma membrane, and that just helps to provide a little bit of support so that it can maintain its shape. And these microfilaments are going to make up your microvilli that are found in your intestinal cells. So microfilaments that are involved with motility will also have myosin along with actin. So we see these in our muscle cells. The actin filaments are arranged in a parallel setup to one another, and then the myosin fibers are going to be between those thinner actin fibers. And you can see when they are relaxed, it allows those actin fibers to spread out. And then when they contract, the myosin helps to bring them closer to one another. And pseudopods are able to extend and contract through that type of motion where they're able to assemble and then connect the actin subunits closer to one another into microfilaments and then able to separate them back out. So I told you we're going to spend a little bit more time with cytoplasmic streaming in our photosynthesis lab, but there you can kind of see an example of what's happening. When you see this, you will actually see the physical movement of materials throughout those plant cells. Actin-myosin interactions and also sol-gel transformations help to drive that process. So intermediate filaments, again, are going to fall between the microtubules and the microfilaments. They, again, assist with holding those organelles in place, and they are a little more permanent than the other two. So when you have your intermediate filaments, they're going to be more static. And then the final part that we're going to talk about is the extracellular components and how cells are connected to one another so they can coordinate activities. Cells synthesize and secrete materials that are going to need to leave to exit the plasma membrane. Ones that are going to fall in that category include your extracellular matrix that's found in your animal cells, cell walls that are found in plants, and then also cell junctions that connect cells to one another. Cell walls, as we've talked about previously, are not found in animal cells. They are found in prokaryotes, fungi, with some protists. They help to provide protection to help to keep the cell shape, and they also play a role in the water balance that's present in the cells. These plant cell walls are made of cellulose fibers that can be embedded in other polysaccharides and proteins. There can be lots of layers to them. You've got a primary cell wall, a middle lamella, and a secondary cell wall. Primary tends to be a little bit thinner and flexible. The middle lamella is a layer between the primary and the adjacent cells. And then the secondary cell wall is between the plasma membrane and your primary cell wall. And there are channels that can be formed between adjacent plant cells called plasmodesmata, and we'll spend a little bit more time on them later. The extracellular matrix is found in our animal cells, and basically it's just surrounding the cell membrane of our animal cells. It consists of glycoproteins like collagens and proteoglycans and fibronectin. And extracellular matrix proteins are able to bind to receptor proteins using integrins. Extracellular proteins that bind to receptor proteins are called integrins. So when you have connections between that matrix that's right outside of the cell and the cell membrane, it's those proteins that help to connect the two together. The matrix is able to help with support, it's able to help with regulating, it's able to help with movement, and it's able to help with adhesion, sticking to one another. So cell junctions that can form between cells and tissues or organ systems have to have a way to interact with one another. They can either be adhered to one another, they could be close to one another, or they can communicate through direct contact. And I said we were going to come back to plasmodesmata in a second. And so plasmodesmata is one way, tight junctions, desmosomes, and gap junctions are other ways that cells can communicate with adjacent cells. The plasmodesmata are only going to be found in your plant cells. These are channels that break through plant cell walls. This allows water and some small solutes, occasionally some proteins and RNA to move between those plant cells. Your tight junctions, your desmosomes, your gap junctions are going to be found in your animal cells, and you can see examples of each of those below. Tight junctions are when your cells are pressed close to one another so that extracellular fluid does not leak. Desmosomes are more like anchors, and they help to connect cells together in as a stronger unit rather than having individual cells. They're more multiple cells working together as one unit. And then gap junctions are going to help cells communicate via the cytoplasm.